Ned Wingreen, who will tell us whether cross-feeding is evolutionary stable.
Yeah, hi, everybody. First off, thanks to the organizers for getting a chance to talk at this amazing meeting. I've been super impressed by the talks so far. And in fact, many people have managed to actually go from models to data and back. I'm going to tell you today a very simple story. It's a work in progress. I think we've gotten models. I don't think we've gotten to data, let alone getting back. But I'd love to chat with people afterwards and hear thoughts about how we might actually make it all the way. So this is work I did in collaboration with Bo Liu, who's now a graduate student at Harvard, and Jiwan Lee, who's now running her own lab at Peking University. So our motivation for thinking about this was in large part this experiment that was done by Alvaro Sanchez, showing that cross-feeding can be stable in the laboratory. It may not be the only experiment along these lines, but it was pretty nice. And Michael Tikhonov will answer all questions about this because he was an author on that paper. I'll tell you about it, but then I want to transition and tell you about a simple model that that allows for cross-feeding. And we were able to analyze this model because it's simple enough that we could actually look at evolutionarily stable states and then ask the question of who gets to coexist if cross-feeding is possible. And sort of the take-home message, I guess, from this short talk is that waste products are going to turn out to be important. So it depends not just who eats what and how they compete, but what are the wastes that get produced. So just to review very briefly, this lovely experiment from Alvaro Sanchez and coworkers, they took some, microbiota from nature. There is a leaf and some cells were extracted from it. And then these were grown by serial dilution in the laboratory. So growing them up and diluting them down again. And the key was that there was only one carbon source here. And so in this particular case, imagine that there's glucose is the only carbon source. There's nitrogen sources and phosphate sources and plenty in there. But all of these cells can only get their carbon initially from glucose. And what was found was that after a few rounds of serial dilution, when various things happen, some taxa go up, some go down, some go extinct, they ended up with a variety of different taxa being present. And this was stable. This was going to keep going after more and more days. And so somehow, despite the fact that there was only a single carbon source, and so in sort of the most naive version of competitive exclusion, where you can't have more species coexisting steady state than the number of nutrients, there seems to be some violation of that. And of course, the answer is that it's some kind of cross-feeding, that while the original carbon source is glucose, the cells process the glucose into other carbon-containing molecules. These are secreted or leaked out of the cells, and they become available for other cells to consume. And then in a separate experiment to demonstrate this, you could see that by growing up particular taxa by themselves on glucose and then asking, having then depleted the glucose, could other cells grow on this residual material? There turn out to be a lot of these arrows, which means that, if Pseudomonas is grown up on glucose till all the glucose is gone, and then that supernatant is extracted, Citrobacter could then grow on that. So there's something other than glucose in there that these cells can grow on. So this paper made the really nice point that, that not only do microbes condition their own environment by consuming things, they also produce things that other bacteria can then consume. And so our thought was, okay, well, this is interesting. Good, cross-feeding can be ecologically stable, which is clearly a very important idea, but there's no time in these experiments for these cells to evolve. There's no chance, for example, for Pseudomonas to learn to eat all of these additional waste products and maybe take over and leave nothing for the other cells. So the question is, could cross-feeding also be evolutionarily stable? Now, we're theorists and we also don't want to spend years doing these experiments. So we decided to try this on some simple models on the computer. And so here I'll explain what we did, which is really embarrassingly simple, but I'll tell you about it anyway. To answer this question in a very simple model world, is cross-feeding evolutionarily stable? And so we imagine sort of a very simple setting, a chemostat, where the cells are going to be in here, nutrient is going to be supplied. We're going to, again, consider the case where there's going to be a single nutrient source. Think of this maybe as glucose again. The cells grow up here, consuming the glucose, and are diluted out continuously with cells coming out and whatever's left from the nutrients. And here is, our generic organism, of which there can be various varieties. This is sort of the generic bacterium. And there's a substrate that it can consume. And the idea is that it's going to grow based on a couple of chemical reactions. So there's one where substrate is converted by some enzyme into an intermediate, and that gives you something, say ATP. But then there's a choice, and depending on the species of bacteria, it could actually process this intermediate another step into some final product and get more energy out of it, or it could leak this out directly and just take what it could get from this first step. And the idea is, well, why wouldn't you do all of these things? And the answer is, well, because you have some fixed budget. You have this enzyme and this transporter and that transporter, and there's no free lunch. You get only to have a certain amount of protein, and so there's a sum rule on these enzymes. So you could imagine that if you divide it up, well, maybe it would be better to have all your enzyme at the first step, or maybe some and leak out something, and that would then be a waste product for you, but that could, of course, be food for another species. So this at least has the potential of allowing for cross-feeding. And so I'll kind of quickly go through the equations, which are not particularly interesting, except that I want to point out that we don't assume any problem per se with toxicity of intermediates, which has been studied in the past, what happens if you have toxic intermediates. But we do take into account thermodynamics. That is, that if you have a buildup of this intermediate, that slows down the rate of this forward reaction. And so here in our equations for the fluxes, we have not only the forward term based on, say, the substrate, but also a reverse term based on the buildup of intermediate for both of these. And so we're sort of, the only thing we're taking into account as far as the problem with building up intermediates is product inhibition. But you can think of this as a kind of toxicity. That is, if you build up a lot of this intermediate, it stops your ability to get energy out of this reaction. And so you can think about the degree of toxicity having to do with the inverse of this equilibrium constant for that relation. If there's no tendency to reverse, if that overall factor is 0, then intermediates are fine. And if it's not, then there's some degree of toxicity. Okay, so there are some equations. We balance our fluxes. The growth rate is just going to be the total amount of ATP you get summed out of both of those reactions. And then the cells grow according to their growth rate minus the dilution rate in the chemostat. You can trust us at least that we can do the math on this and then see what happens. But now it's simple enough that we can start asking, well, instead of just throwing in a few different random choices of species, we can ask what kind of combinations would be evolutionarily stable. And so we can sort of show generally that there are only a few possibilities based on this sort of generic bacterium. There's something we call the polluter, which just does the first step. It's like, okay, I'm going to get energy out of this first step, and I'm going to leak out my intermediate, and that's sort of going to build up this kind of toxin in the environment. You could have something that goes all the way, that says, well, I'm going to get energy out of both steps, and I'm going to then pollute the environment with the last possible product. But then where there's for the polluter 1, there's also a cleaner. This one says, look, if polluter 1 is around producing this intermediate and leaking it out, I can just use that as food. And so clean things up and get energy out of the second step. And in principle, there's something that could do everything. It could sort of have some amount of enzymes and transporters devoted to everything. So what do we find? So we thought, well, here's polluter 1 and cleaner. That seems like a nice combination. Maybe those guys can be stable. And indeed, they can form a cross-feeding pair. If you put in polluter 1 and the cleaner, and fix their strategies, they happily coexist in this system. But is that evolutionarily stable? In other words, can those last in the face of invasion by other alternative strategies? And there, it turns out the answer is no. You do get coexistence in this system, but it's not between the polluter and the cleaner that would have sort of been, I eat something, make a waste product, you eat my waste product, clean it up. Seems good. But what actually happens is you get coexistence between the two polluters. That is, as you add more nutrient, the first cells that can exist against dilution are the polluter 1. They put all their enzyme budget into that first step, get as much energy out as possible, and pollute the environment with the first intermediate. But as they do that, and their population goes up, which is this color here, they make the environment more and more polluted, which effectively slows down their ability to get energy out of the substrate. And then at the steady state, the substrate level creeps up to the point that now the other thing that can survive is polluter 2, that takes in that somewhat higher level of substrate and runs it all the way through to finally produce the final product. But it doesn't actually get any toxicity from the intermediate because it doesn't exchange with that intermediate with the environment. So you get coexistence in this system, but not coexistence of cross-feeding. It's coexistence of guys who pollute their own environment to the point that they start allowing other species to coexist with them. And so it's these guys, polluter 1 and polluter 2, that get to coexist. Turns out that if you play with the parameters, you can go the other way, where polluter 2 becomes the first one that's able to survive as you increase the nutrient supply, and then polluter 1. And that depends on the details, like the degree of dilution and the amount of toxicity of the intermediate. And of course, you can produce phase diagrams here. The point of these phase diagrams in terms of nutrient supply and dilution is that you never get coexistence between the polluter 1 and the cleaner. The things that would have thought of as the simple version of cross-reading is never evolutionarily stable. All you get is combinations of different polluters coming in at different points in this phase diagram. So what's going on? Well, it turns out that this is sort of generic. If you look at longer pathways, you get the same story. If I have other intermediates, all I can get is coexistence among different polluters. This one who pollutes with the first intermediate, this one who pollutes with the second intermediate, this one who pollutes with the third intermediate, and so on. You never get a situation where you have any of those cleaners that would use these intermediates as their food source. Those are never evolutionarily stable. And so why is that? And it turns out that you can understand this kind of mathematically. You can say, well, look, if I actually have the best possible values for a polluter 1 and a cleaner, so that these come into some perfect case of all possible polluter 1s and all possible cleaners. These are the best combination. What you end up with is basically a megacell that's a polluter 2, right? It does the whole reaction all the way from here, getting energy out of there, takes this intermediate and gets energy out of there. It exists as if the whole chemostat was one big cell that was doing both steps. Only in this case, they actually have to pay some cost for transport. Whereas if you really had it in one cell, you wouldn't have to pay the cost of these intermediate transporters. So we can prove mathematically that in fact, the best possible combination of polluter 1 and cleaner is never better and always worse than polluter 2. So that's why we never get real cross-feeding in this model. But it does raise the question of like, well, what about this case of making alternative waste products? I mean, that's what we're finding coming out of this model, is that there are different strategies having to do with what you make as a waste product. And so yeah, this is just a summary of what I just said. Well, alternative waste products is a real thing. This is just a super simple picture that I stole it from Wikipedia, but glucose comes in and you can get energy out of it by fermentation, but there are various possible final fermentation products, like lactate or succinate, acetate, ethanol. And, I've always thought of this as just well, okay, somehow evolution ended up giving, yeast makes ethanol, E. coli makes, lactate and acetate. It seemed kind of random. But now you're thinking about it, it's like, well, if these are the waste products, those are then feeding back to particular pathways. But by having different waste products, maybe you can make your pathway more efficient if, for example, you choose to make a different waste product than the dominant species around. So the question is, can that work in the sense that what if you could only get energy from this first step? Would it still ever be worthwhile to run this second step so that you produce a different waste product? It seems a little weird, like, oh, why would you run extra metabolism here that you're not getting any energy out of? But it turns out that this business of accumulation of waste product actually can make that worthwhile. And so here we have a case where you have now, as a function of dilution rate, coexistence of two different polluters. Polluter one makes sense. It's the one that does the most efficient reaction and makes the waste product. But polluter two is actually running an extra step in metabolism that gets it no benefit, but accept the fact that it actually produces a different waste product than polluter one. And so by polluter one is present and polluting the environment with that first intermediate, it opens up a niche for polluter two that manages to spend extra part of its enzyme budget running extra metabolism, but as a result has a final product that has not accumulated in the environment due to the presence of polluter one. So this for me was actually surprising. I had not recognized that there would be a pathway that would be useful, even though it didn't generate any additional energy or growth for the cell, just as a result of avoiding dealing with a pollutant that would build up in the environment. So, okay, different strategies can produce coexistence that way. So that's it. I'm actually on time. So to summarize, of course, microbes do create their own environment. I guess I'm preaching to the choir with this group of people, but I think this is a point that's always worth making. In this very simple model, cross-feeding can be ecologically stable, perfectly fine to put in polluter one and cleaner, and they get along great. But if we allow evolution, then this is never stable, and we end up only with this combination of polluters. And then, for me, an interesting result of this was that different polluters can not only coexist, but they can actually engineer their metabolism exactly for that reason, not to get energy, extra energy out of additional steps, but just to avoid making the same waste product that everyone else is making. And so with that, I will acknowledge my coworkers on this, Bo and Jiwan, our funding sources, and I will open it up for questions.
Awesome. Yes. So thank you, Ned, for being